All right, thank you everybody for coming, and thank you for those that might be watching this on Zoom at a later date. It is my great pleasure to welcome Zhuzhi Zhang, who has done his master's in our lab, looking at the effects of water on seed beetles. I'm not going to tell you about that because he's going to do that. But I just wanted to give a little brief introduction. So Zhuzhi came to our lab 2 years ago, I think, pretty much, and was here for a few weeks and then left again. He had visa troubles, so then ended up going back to China and came back to Canberra a few months later to actually get started with his master's. It wasn't smooth sailing at all. The first field season was, he was going to work on a different project, but we had bushfires, hail. Yeah, lots of things went wrong. Nothing to do with Zhuzhi's abilities at all. In fact, he coped superbly well with all the difficulties and moved quickly into another system, and didn't look back after that. I'm sure that Zhuzhi has had, I don't know, he could probably tell you better the kind of, I don't know how to explain how to say it, but you know, there are a lot of cultural differences, I guess. And so he will have, I'm sure, learned a lot about Australian culture while he's been here. And I've certainly learned a lot about Chinese culture since he's been here, which has been really nice. Yeah, I guess it's great to see that you've come this far and that you're finally in the throes of writing up and getting everything done. You've conducted an enormous amount of work, which you should be super proud of. So with that, please take it away.
Well, so hello everyone, welcome to my Access seminar. Today I will introduce the work I have been doing for my master's over the last 2 years. So resources intake is an essential process for organisms. Individuals have to absorb nutrition for their metabolism and development. However, sometimes nutrition may not be enough. And in this situation, fitness of individuals is inevitably affected. And besides of obtaining enough resource, different requirements in different life stages is also important. Besides the original resource carried by individuals, it can affect offspring body mass and their development rate. Juvenile resources are allocated into development and also stored for adulthood. And the adult nutritional ingestion can be allocated into reproduction, but at the same time, it can also be used to maintain their body and prolong their reproductive age. For example, when large cabbage butterfly larvae develop under high predation pressure, they have to allocate more food into their regurgitate defense. And they consequently have lower survival, smaller pupae, and less eggs. Besides, limited nutrition in male scorpion fly larvae not only restrict their adult body weight, but also interact with their adult diet to influence the salivary gland weight relative to their body weight and further influence their mating success. Besides of food, water is also an important resource for creatures to survive and reproduce, especially for animals living in dry environments. However, compared with food, exploration of the effect of water on life history is still limited. To step forward on this topic, I choose the seed beetle as the model species to carry out my research for several reasons. First, their larvae develop within beings and cannot move between them, so it's easy to manipulate their developmental environment. And second, they occur on stored grains where water is limited. And third, water affects the economic of mating in this species. Water conditions of early life stages have the potential to last into reproduction performance. And during my master, I looked at how access to water at different points of life affects several aspects of seed beetle life history. First, I looked at how it affects maternal oviposition preference. Here, females were provided with wet or dry beans. And you can see it's wet or dry beans. And then I recorded their preference and measured their offspring's performance. And of course, on both type of beans. And second, I looked at how male and females' access to water during development affect mating behavior and female fitness to determine the role of water in sexual conflict. And third, I manipulated the access to water during development and adulthood, but this time only on males. And it's to look at how access to water affect male investment in reproduction throughout their life and determine the relative importance of developmental versus adult environment and whether there were any interactive effects. So following the time sequence of their life history, let's start from the oviposition preference experiment. As we know in human, adequate nutrition is essential to have a healthy baby. The same in insects. Not enough nutrition can cause delayed development or poor body condition. And these disadvantages can last into adulthood and may even reduce the ability of individuals to reproduce. In this situation, where mothers lays their eggs could be important for the next generation, especially in those species whose larvae have limited moving ability, like seed beetles. They are very good example of a species with developed in a resource-limited environment, because each larvae was restricted to a single being, and these beings are always very dry and lack of water. Like for almost every living creatures, water is a precious resource of them, which can affect lots of traits in all life stages. So for female beetles, being able to distinguish between beings based on their water content could allow them to improve the performance of their offspring. This experiment was focused on testing this ability on water and measure the fitness of developing in wet or dry beans for offsprings from these conditions. To test the preference of wet or dry beans, I have to manipulate the water content of the beans. This was done by storing the beans for 24 hours in small containers within a larger box that had a wet toy in it. Control beans were treated in the same way, but without the toy. To check this method was effective, I weighted the beans before and after being placed in the container. After this treatment, wet beans have gained around 1.6% of weight, and dry beans had gained no weight. And these beans were later provided to mated females to test their opposition behavior. To have a more comprehensive view of this question, I used two different tests. So it's two choice test and no choice test. And in two choice tests, female beetles were provided with both wet and dry beans at the same time. And they can choose the beans in a better condition from many beans they can access to at the same time. Mated females were allocated into two choice tests for 2 hours, and then they were moved into no choice test. In the no choice test, females were firstly allowed to lay eggs on one type of beans for 1 day and then transferred to the opposite type of beans for another day. This test was set to test the preference when females can access to the better condition, when they can access to the better condition where they have access to. With these beans, I counted the number of eggs females laid on each type of beans, and also measures the development time and the emerged body weight of their offsprings. For the result, this figure shows the egg laying preference of two types of tests. The red dots correspond to the two choice test, and the blue dots correspond to the no choice test. Let's show here, maybe it's too small. And And the x-axis here is the number of eggs a female laid on wet beans, and the y-axis is the number of eggs laid on dry beans. So we can see that the dots are evenly distributed on the both sides of the no preference line, which means that overall females show no preference for laying their eggs on either wet or dry beans. And this result was the same regardless of the type of the test. Meanwhile, offspring traits didn't show any difference between wet or dry beans. Okay, so although the importance of water in seed beetles has already proved from various aspects, the result still shows no preference of beans with higher or lower water content. So for the reason of this phenomenon, I have several potential explanations. First is beans absorbed water may expire a kind of deterrent to prevent female beetles from laying eggs on them. This assumption was supported by research on black-eyed beans. They detected D. lemony is a kind of famous insect deterrent from wet beans. And there is still no direct evidence on mung beans, but it's reasonable to assume the same situation on this kind of beans. And besides of it, females may don't have the ability to distinguish water content of beans and focus on the evolutionary aspect, as there is no developmental consequence caused by wet development environment or beans here. There's no selection pressure for the females to have the ability to distinguish wet beans, but the wet developmental environment do affect adult performance, which may also affect fitness of beans of beetles reared in wet beans. And this argument will be discussed in the next part. And so my second experiment was focused on how water obtained by males and females during development affect their mating interactions and adult fitness. And sexual conflict occurs when two sexes have conflicting optimal fitness strategies concerning reproduction. And it's well-spread and common from mammals to birds and insects. And it also has various forms like infanticide, repeat copulation, and transferring toxic ejaculate chemicals. Sexual behavior and strategies are often affected by the environment where mating takes place and males or females are reared in. Like in cockroach, where males reared with conspecific females would invest more sperms in their first mating. And in fruit flies, their males adjust their seminal fluid ingredients depending on the population density and on the population density they were reared in and their own body mass. From these examples, it's clear that the social environment that males experience during development can be important for determining the expression of male reproductive traits. Here, I was specifically interested in looking at how access to water during development affects sexual conflict. Specifically, I wanted to know whether have access to a limited resource during development increase sexual conflict, because females may no longer need to mate multiply with males to gain access to that kind of resource. To answer this question, I conducted an experiment where I manipulated water availability of males and females independently during development, and then looked at their mating behavior and fitness. In detail, with beetles that emerged from the eggs of my oviposition preference experiment, I wanted to test how the environment that beetles were reared in affects sexual conflict. To do this, I paired wet and dry beans in all combinations to look at their mating traits. This crossed experimental design allowed me to tease apart whether potential effects of rearing treatment on mating interactions was driven by males, females, or both. After the first mating, males and females were separated through the night and then mated with the same partner for a second time on the following day. By mating pairs twice, it allowed me to look at whether effects accumulate over multiple mating. After pairing beetles, I measured their mating behavior and fitness. The mating trace I measured was behavior, including the time it took females to start kicking after the onset of mating, the amount of time females kicked the male before the mating was terminated, and the total mating duration. I also measured the male ejaculate size. This was calculated as the difference in male weight before and after mating. After mating, I then transferred females to a container with fresh dry beans and let them lay eggs for 1 day, and then measure the lifespan of females and survival of their offspring. For the result, for all figures in this section, the x-axis is the water treatment of paired beetles. The first letter is the water treatment of males, and the second is for females. And figures in these slides are showing the mating behavior result. They showed no difference between groups, and this result was the same whether in the first or second mating trial. And then look at the ejaculate weight on the other hand. We can see that wet males mating with dry females transferred a larger ejaculate than all other pair combinations. And so for this experiment, what do my results tell us so far? Well, first, the ejaculate of the size of the ejaculate of a male transferred depends on both males and female rearing environment. My results suggest that males who have more water during development are able to produce larger ejaculate. But that either they only transferring larger ejaculate to dry females strategically, or that only dry females are willing to accept the larger ejaculate and cause a higher level of sexual conflict. Interestingly, the fact that there were no difference in mating behavior suggests that behavioral changes are not the reason of the difference in ejaculate size. Then I looked at the reproductive consequence. First is the egg number. Although it looks like there could be an interaction, but actually the only difference was between males. And here, wet males had fewer eggs than dry males. And however, the survival rate of eggs showed a different pattern. For females who mated with dry males, for females who mated with dry males, those who reared in dry beans laid more eggs than those who were from the wet beans. And mating with wet females, both dry and wet females laid a similar number of eggs. When I combined the egg number and survival rate, and it's the number of adult offspring emerged. It's the most important fitness measure because it's these individuals that go on to contribute gene to the next generation. So emerged offspring number shows the active and negative effect of developmental water content. In detail, dry pairs has the most offspring and the wet pairs have the least, and mixed pairs are between them. One possibility of the lower number of eggs produced by wet pairs is that wet females received fewer chemicals that promote eggs laying from the ejaculate. Considering with the strategic ejaculation, results suggest that although wet males transferred the same size of ejaculate to wet females as dry males due to other type of females, this ejaculate it may be more dilute because it may have more water and less chemicals or even less sperms. But this argument still needs further verification. Once we look at the number of offspring survived to adulthood, However, we see that dry pairs has the highest reproductive fitness and wet pairs has the worst. This result could occur if beetles are locally adapted to dry environments or having increased access to water during development increase their sexual conflict. And in previous study looking at how water availability during adulthood affects mating behavior and fitness, it was shown that access to water increased female body weight and lifespan, suggests that water is a limited resource. But in my experiment where I manipulated the amount of water during development, wet developmental environment access to affect the size of ejaculate that males transferred and the reproductive success of females. This result suggests that having access to a limited resource during development may increase sexual conflict because it alters the economic of mating. Finally, my results also showed that the effect of rural environment can last into adulthood and even the next generation. So in the experiment I just presented, I looked at how male and females access to water during development affect mating interactions during adulthood. And I found that access to water during development was important in determining ejaculate mass, but not in mating behavior. I also wanted to highlight how this result different from previous studies, which manipulated male and female water accessibility during adulthood. It shows that adult access to water tend to influence mating behavior, but not in ejaculate mass. So for my final experiment, I was interested to look at the relative importance of developmental and adult environments and whether there were potential interactions between them. Sorry. And in the different life stages of insects, the nutrition can be allocated into different years. But they can also affect and interact with other life stages. Like mentioned around the beginning, in scorpion flies, both larvae and adult nutrition shapes their salivary gland and affects the mating success. And also be reviewed by Bogus, larval feeding can affect adult fitness and reproduction throughout life stages. However, the effect of water is still not being fully understood. In the previous research, Vincent have introduced male seed beetles' access to water during adulthood have heavier body weight and longer mating duration. But when I manipulate water accessibility during development, males reared in wet bins transferred larger ejaculate. So to take a closer look at these different effects caused by water, I run this experiment to explore the interaction and the difference of how the water affect males' fitness and mating traits. To do this, I manipulated the water content in the water condition in both development and adulthood. At this time, I used a slightly different method to generate wet bins to match better to what have done by other studies. This time, beans were kept at 4 degrees centigrade for 10 days to give them enough time to gain water and prevent from getting moldy. Dry beans were kept at room temperature in the same 10 days. So here, this time, wet beans gained about 2% weight after the treatment, and the weight of dry beans stayed constant. In this experiment, I focused on the effects of juvenile and adult water availability, specifically in male reproductive traits and how investment in these traits change throughout their adult life. To look at the potential interactions between water availability at different life stages, I used a 2x2 factorial design to produce all combinations of males. Males were put into their water treatment from the day they emerged as adult and then began mating trial 1 day later. Then the males were mated twice a day for 3 days, each time with a new virgin female. This allowed me to explore the effect of juvenile and adult water from several aspects, such as body mass, ejaculate, mainly on ejaculate size, and other fitness traits. Just after males emerged, they were weighted and randomly allocated into wet or dry treatment. Body weight of males can affect so many traits related to mating, like mating duration and ejaculate weight. And body weight is always in positive correlated with their lifespan. So body weight change can affect lots of later traits of males. So here we can see that before mating trial, although all males lost weight, but males can access to water lost more, which is a little bit strange. To evaluate the seminal fluid replenishment potential, I also analyzed the body weight recovery after the first mating. Here, males can access to water recovered more body weight between 2 mating days. And in the first mating, males can access to water during adulthood transferred a larger ejaculate, which was different from our previous results. However, ejaculate replenishment between days and ejaculate weight decrease in each day was not affected by development or adult water treatment. And I also analyzed the mating behavior. Although the mating behavior in the first mating didn't show any significant difference, but in the third day, males from the wet beans was kicked later than males from the dry beans in the first mating. I mean the first mating of the third mating day, which suggests that females mating with these males were satisfied or cannot endure the harm from mating later. And for 2 matings in the same day, mating behavior was not affected by water treatment during development or adulthood. So from this experiment, the ejaculate weight was affected by adult water accessibility rather than rare water condition, which is different with our previous result. Besides, it's strange that males' access to water lost more weight after emerging than those who can't. But in longer time period, access to water helped males to maintain their body weight, but water they gained seems did not mainly allocated into ejaculate, as access to water did not help them to replenish their ejaculate, which is consistent with the previous assumption. So from all works I did in my master, we can know that development and adult environment do have different effects on mating and reproductive performance. Redundant water gained as larvae and adult was allocated into different use. Although there are some results are not consistent, my experiments still provide evidence about the effect of water, especially the effect of juvenile water availability. And my experiments are also proved that similar to other nutrition, water acquirement in different life stages may also be allocated into different functions and further affect adult fitness and reproduction from different sites. In the next step on this topic, it could be to look at the physiological mechanism of the difference of water effect during development and adulthood, and explore the ingredient difference in larger ejaculate, or to look at the relationship between ejaculate size and mating duration, questions like, do larger ejaculate was transferred for quicker or longer? And at last, I would like to thank many people. First of all are my supervisors, Megan, Michael, and Dan, and especially Megan. Without her help, I can never do this experiment and finish my paper. And then I would like to give thanks to my girlfriend. She supported me so much, not only on my life, but also gave me so much help on my experiment and be the first audience of my talk. And I want to give thanks to people of Head Lab. You are amazing colleagues and always so happy and active. And to all my friends, especially, and also give thanks to all my friends, especially Ophia and Ian. And they gave me lots of help when I was crazy with my experiments. And last, thanks for all people in us ISB. You built up a so great department.
做得好，那是个好话。
Can you do that? I don't understand that.
I hope it was correct.
Yes, it's correct, but I can't remember.
I'll say it again for you.
做得好，那是个好话。
All right, so
Well done. It's good.
oh, fantastic! Well done. Yes. All right. So thank you, Zhuzhi. That was a wonderful talk. Does anyone have any questions for Zhuzhi? I'm going to jump out of the way. But we might need to repeat your questions for the.
Question about your second experiment. So let me just repeat if I remember correctly. So males in wet conditions produce more sperm when they mate with females in dry conditions, right? Said that,
but actually, they transfer larger ejaculate,
yeah.
but we don't count the sperm number, so we don't actually know.
That's what I was wondering, if there's a way to tell if they're just transferring water, like because you mentioned that protein or like chemical content might be important. Is there a way that you can do sperm count?
Yes, there's some way we can do that. Like for me, the most hard part of this is to extract their ejaculate. But after that, with their ejaculate, we can like with the dry weight of their ejaculate, or there is some way to count the number of sperms in their ejaculate. Yeah, but my plan, if I have time, I will do that. But I can't remember it, and I don't know the exact way how to do that. But I saw several papers talking about how to count the number of sperms. Usually beetles, this kind of beetles, they don't drink or eat in adulthood, but if they have chance, they can, and they want to drink something, or if there's some nutrition in the, like some sugar or things in the water, they can take it. But usually they don't drink or eat anything. All their nutrition are from their larvae. And any other questions?
I just have a question on the first experiment. So in the first experiment, you implied that females are and I might have missed this, but as one of your explanations, you suggest that females are here to reconcile the explanation. Can you explain that? Why would females not like the needs, but the results? Yes.
Okay, yeah. Actually, this experiment was a part of my second experiment, so I didn't look at it very close to the experiment. But from other experiment I mentioned, they did the similar thing in black eyed beans and find some deterrent from them. They do different things, and they found beetles prefer larger beans, and when they cover the surface of beans with something and make females can only touch to a small surface of the beans. I mean, Sorry. And when they are doing that, they found females do prefer. Sorry, it's a little bit strange. I will say it again. They found females prefer larger beans, but usually when beans get absorbed water, it become a little bit larger, and maybe it's a reason why they suppose it's a reason why females prefer larger beans. Sorry, and when they cover the most surface of the beans and only left a very small area for them, they don't show any preference. And in my experiment, I guess it may be because the weight difference of wet beans is not that huge, so females didn't show any preference. And no end. Yeah, that's all. Sorry.
All right. Well, I think we might let Zhuzhi go now. I think he's answered enough questions. We're not having a big celebration today, but I do have some champagne and snacks if people would like to join us now on the terrace